Hello, my name is Dr. Bradly Alicea, and I'm the head of the DevoWorm Group. And I'm going to tell you a little bit about our history. And this is our 10th anniversary, as you can see. And this is 10 years of computational developmental biology. So I'm going to tell you a little bit about DevoWorm and our history and what we do and what the future looks like for us. So we're a distributed group of scientists, computer scientists, biologists, other types of physicists, biophysicists. So we're a distributed group of collaborators. We have biologists, computer scientists, physicists, biophysicists, and engineers. And we're interested in simulating the development of C. elegans, but also the development of other organisms as well. We have a presence on GitHub, Hugging Face, YouTube. We're affiliated with the OpenWorm Foundation and the Orthogonal Research and Education Lab. And we do our meetings on Jitsi. So we have a number of tools that we use to collaborate, and we have a number of affiliations with which we work with larger groups of people on different topics in modeling and simulation, as well as some biological problems as well. So in the beginning, we started in April of 2014, so we're right around the 10th anniversary. We started with this issue. So we actually started our group not with a project vision, but with answering a call for someone to address a GitHub issue in the OpenWorm Foundation. So this is Steve Larson, who created this issue, number 179. And the issue reads, add cell lineage info to PyOpenWorm. So this is something they had going on at the time called PyOpenWorm. And it's a Python tool that, toolbox for sort of unifying OpenWorm, which at the time was a series of toolboxes to simulate the nervous system of C. elegans, simulate the cells of C. elegans, simulate some of the biophysics and the electrophysiology. And so that's what they were trying to do. And the reason why you would want to add cell lineage data to PyOpenWorm is because in development, we know in C. elegans what every developmental cell will become in the adult. So it was very useful for their end because they're largely interested in modeling the adult worm. Very interesting to them to have some sort of developmental lineage associated with the cells in the model. And so this is something that we didn't actually end up addressing, but that's fine. We became our own project. The other reason why you'd be interested in developing a model of a cell lineage for an organism like C. elegans is because in C. elegans that we know what the cell lineage tree looks like. It was worked out in the 70s, and we know kind of what those cells will become, and we know the timing. It's very well characterized. And so, as well as the adult being this kind of eutelic organism. And when I say utily, I mean that the adult has a fixed number of cells, unless it's a mutant, has a mutant genotype. So the number of cells is never variable. And this is very fortuitous for modelers because you don't have to deal with a lot of variation. Although in our group, we've dealt with models that are sort of interested in looking at biological variation. But that's for later on in the talk. So our first meeting was on April 11th, 2014. Our attendees were Stephen Larson, myself, and Richard Gordon. He's a retired developmental biologist. Steve Larson, of course, runs MetaCell, which is a biological modeling and neuroscience, neuroinformatics type outfit, and they build a lot of tools for biological modeling. And so we met together in Google Hangouts. That's how long ago it was. And we kind of, got a feel for the landscape of the problems. And that first issue was kind of malleable because, that was something that they wanted to do. They wanted to incorporate some basic developmental biology into their modeling scheme. And of course, as having the background that Richard and I do, we suggested other ways to sort of fulfill that role. and that involved a lot of sort of biological modeling, biophysics, and developmental biology. And so we decided we were going to start this project and this collaboration and see how long it would go. And I don't think any of us thought it would last 10 years. So I told you a little bit before about modeling the nematode C. elegans, how amenable it is to modeling because there isn't a lot of variation in the phenotype, in the developmental phenotype. We have a well-characterized lineage tree and so forth. Now, a lot of groups, aside from OpenWorm, have tried to model different aspects of C. elegans, build virtual C. elegans, build the perfect C. elegans, and so forth. So this is just a sampling of the different initiatives that have popped up around C. elegans. So at the top, we have this PLOS One paper called C. elegans, S-E-E elegans. C. elegans, simple to use, accurate, and automatic 3D detection of neural activity from densely packed neurons. So this is a focus on the C. elegans connectome, which we have knowledge of every cell in that connectome and every connection in that connectome. So we can do things like this. We can try to build models that are sort of hyper realistic and, get a fix on the biology and, do that, or we can build models based on data from those kind of initiatives to, do different types of simulations and see what happens if, have these hypothetical scenarios for exploring the functional biology would be a good way to put it. Another project is the C. elegans, and that's SI, though, so that's like yes in Spanish. The C. elegans project. The challenges and prospects of emulating Cantorhabditis elegans. They did this approach called emulation, which was, creating maybe like a digital worm, modeling it very hyper-realistically, and then putting it to the test and seeing if they can replicate, everything about it. So that's another approach. There's also MetaWorm, which is a newer paper. Now, we covered this in one of our lab meetings. An integrative data-driven model simulating C. elegans brain body and environment interactions. So in this paper, they've developed their own tools where they've integrated a lot of things to sort of build this this closed loop sensory motor system, and they're simulating movement, but they're also simulating some interactions between the body, the brain, and the worm's environment. So there's a lot you can simulate, not just the biology, but you can also simulate sort of the embodiment of the worm as well. So their paper at the top, Towards a Virtual C. elegans, a framework for simulation and visualization of the neuromuscular system in a 3D physical environment. So this is actually Andrei Palyanov and Steve Larson, Sergei Karulin. These three people are in OpenWorm. They work on biophysical models. They work on the cybernetic model, which is where the worm crawls through different media and the interactions that occur from that media, how it affects movement, and so forth. So, getting these hyper-realistic models are maybe not necessary if you can mimic the sort of biophysical interactions going on. So there are different ways that we want to model C. elegans. There's also the Grand Challenge, full reactive modeling of a multicellular animal. That was at a conference, Hybrid Systems, Computation and Control, back in 2003. And then back in 1998, there was the Perfect C. elegans project, an initial report. This was in the journal Artificial Life. So you can see even back to around the year 2000, people were thinking about modeling C. elegans and, doing so in different ways. Thinking, talking about using adjectives like perfect, meta, virtual, and, different types of hyper-realistic or emulation, different types of hyper-realistic simulation, or, simulation based on some subsystem. So my point of this slide is to show that this is a very rich area of activity. And so when I show you the things that we've done in our project, I want you to keep in mind kind of the sort of approaches that people have used. Now, one thing to note about all of this is that these are projects that focus either on the adult worm or the larval worm, and I think mostly the adult. And I think MetaWorm might talk. They might talk a little bit about the larval stages of the worm, but nobody's really focused on modeling C. elegans embryogenesis. And so that's where we kind of come in with that. And so this was our first public announcement in June 3 of 2014. We had our core members. We had Mark Watts and Steve McGrew, who also joined at this point. And so we were ready to kind of get going on some things. So we had a blog post that laid out what we wanted to do and talking about, our connection to OpenWorm. And we ended up with a project white paper where we kind of laid out a simple informatics model of development. And so our first publication was on October 3, 2014. This was this bioRxiv paper on DevoWorm differentiation waves and computation in C. elegans embryogenesis. So we introduced this theoretical concept called differentiation waves. This is something that Richard Gordon has worked on for a lot of his career. We have an element of computation in C. elegans embryogenesis. So we're trying to model C. elegans embryogenesis from the standpoint of a lot of those papers in 2 slides ago. And so that's where we kind of were going. And to start off with, we proposed a 4-dimensional model for the C. elegans lineage tree using an RDF framework. And the RDF framework is a tabular data structure. And so what we did was we produced a 4-dimensional model, which was spatial and temporal, and kind of built something that you could use to characterize C. elegans development, but also plug it into some of those other types of modeling initiatives of the adult worm. Well, that wasn't our stopping point, fortunately, because that was, I think, a decent first pass at kind of what we wanted to do, but it wasn't really, it was kind of a thing where what they call seed paper, where you put your ideas out there and then you move on and you try to build on that. So that paper, Differentiation Waves and Computation, was on the bioRxiv. We did some OpenWorm journal clubs to sort of introduce the idea to people. Then I visited Indiana University and their Biocomplexity Institute, which was like the group with James Glazier and some other people doing work with CompuCell 3D. And this was something that we wanted to do. We've been wanting to work with CompuCell 3D for probably about 9 years now. And that was kind of where I went to see what they were doing and see how we might be able to use those kind of models. And it really did, although we've never really done very much with CompuCell 3D, it's really informed the kind of modeling we've done since then. We've done some more journal clubs. We did, in 2016, a number of things. This information isometry paper on the bioRxiv, which was analyzing lineage trees using different forms of information theory. We did this paper called Quantifying Mosaic Development, which was a comparison of C. elegans embryo with the C. squirt or the Ciona intestinalis embryo, both of which have this quality of sort of this regularity in development where you know the lineage tree quite well. And so for both organisms, we have this lineage tree that we can work out very easily from existing data, and then transform that into what we call a differentiation tree. And then compare those differentiation trees to see what kinds of insights we can make. So this was published in the journal Biology. There was also some work on cellular automata modeling, and this was something that Tom Porteges, who had joined the group by then, worked on, and we all kind of worked together on sort of this paper called Morphozoic, and the platform was called Morphozoic, and it was a book chapter on this topic. So Morphozoic is a cellular automata that is nested, so you have this grid of cells, and there's spatial nesting of those neighborhoods in that grid. And you can, use different types of, manipulate those neighborhoods in different ways to produce different shapes. So, you have things like you can simulate gastrulation or basic geometries. And so this is, this is just kind of a screenshot down here. It's hard to see on the slide, but there was basically a proof of concept paper. Now we also didn't really drive Morphozoic forward very far, but we're interested in kind of reviving that, as we are with any of these things that I show you. If you're interested, if you don't think we've gotten enough out of it, please by all means try to take ownership of some of it, because it's kind of there and it's open source, and that's the spirit of open source. So in October 2016, we had the OpenWorm Open House, and that was an event where everyone in OpenWorm came together and shared their projects, updates, and I shared an update in 2016 on DevoWorm, but we also had a tutorial on Morphozoic. Then in November 2016, I did some work independently of the DevoWorm Group on experimental evolution, using C. elegans to build an experimentally evolved line of different genotypes of worms. So, we had a wild type worm, we had defined mutants, and then, looking at if you evolve them over a number of generations, what the outcome is. And the focus was on fecundity and how how energetics affects that, how other types of growth mutations affect that. So this is stuff, this is experimental biology that's related to embryogenesis. And this is something, again, it's not a major theme in our group, but it's something that we've done. This is an event in 2018 that happened at the Royal Society. It's called Connectome to Behavior. And so DevoWorm was sort of tangentially involved in that, but that was, I think, a watershed moment for OpenWorm, because not only did we get a special issue out of it, but it opened up our project to to an audience in London, to an audience of different scientific labs, and I think they were really impressed with what we were doing. That momentum, I think, kind of got upended by the pandemic, but I think that was a big milestone in both the DevoWorm Group and the OpenWorm Foundation. In 2018, we also, I presented some work on something called embryo networks at Finding Your Inner Modeler at the University of Illinois Chicago. That was a conference where you had biologists and modelers and people who were kind of hybrids, and they present on their work. And this was an approach that I was developing this is kind of on my own, where you're characterizing those embryos. We had the RDF model in our 2014 paper, and now we're saying, okay, well, we have spatial information, we have temporal information. We can also build connection type information, or like affinity type information for different cells, and then we could build networks from that. So that's what that work was about, which would get published later that year in Biosystems. 2 papers on that, one on sort of embryo networks and another on developmental connectomics. So this paper down here, this was a Proceedings of the Royal Society B paper that was published, sort of an OpenWorm Foundation paper that came out of this Royal Society meeting, and that features DevoWorm in it circa 2018, so you can read more about that as well. Then in April 2019, I was part of a cohort in the Mozilla Open Leaders Project, where they train people to work what they call work open. And out of that, I developed this DevoWorm OpenWorm curriculum. It's something that carried over to some of our work in 2019 on later 2019 on DevoWorm ML, which is like looking at the intersection of machine learning and developmental biology. So we have this educational component in DevoWorm. We had some other papers come out, between 2018 and 2020. This is just a snapshot from 2014 to 2019. This is a 5-year report. So we had a number of papers come out during this time. We had a number of meetings. We had a number of journal clubs within our community in OpenWorm, and a number of educational initiatives. So from the last slide, you saw that there was a logo that looked different from the logo that was on the first page of the presentation. And so we've had a lot of logos throughout the years, and they've been various logos for various purposes. So we started off with a couple of different types of logo, and we kind of evolved that to the present logos that we have. So we went from things like this to this is showing the affiliation with the Orthogonal Research and Education Lab. This is showing our affiliation with OpenWorm. This is kind of showing sort of a quasi-embryo in the context of the different themes that we had at the time, which was around 2017, 2018. And then today we have our present logo, and we also have a logo for DevoWorm, which is sort of our machine learning and deep learning and graph neural network arm of the project. So we have a number of logos. If you see these, they're, some of them are more prevalent than others, but that's what they look like historically. So if we go back to this model, we started with this 4-dimensional RDF model, and it's basically kind of an annotated table, which is sufficient for some informatics applications, but not necessarily sufficient for heavy-duty modeling, where we want to incorporate as much information about the embryo as we can, and also make it easy to take those variables and parameters out and build models based on secondary data, but also on simulated data and the outputs of other things. So this is an example of how we can take secondary data, turn it into a computational model, and then apply it to some domain. So this is on the left, this is a 3-dimensional point cloud. And this is from cell tracking data. And one of the standard data sets in C. elegans embryogenesis is from Murray et al., and that was published in Nature Protocols in 2006. It's been a while since that was published, but the data is probably the best that you have. Or just, there probably doesn't get very much better, because we're not really, in this case, we're not really interested in cell tracking, although you can extract that from this data set. But we're just interested in a point cloud. So we have this point cloud, which is not differentiated according to time. It's just all the cell centroids that exist in the embryo. So you can see it's pretty messy, but it's a appreciable point cloud. You can see some variation, of course, in the spacing of it. So they're clumps of cells, and the trick here is to sort of have that 3-dimensional position data. So you have 3 dimensions of volume, and then you have a time parameter, so you know what time that cell is born, what time it dies. So at any one time, any subset of this point cloud exists in a model of the embryo. There's also something like a migration angle, which is theta, and that's basically where we're looking at a cell that divides. How, what's the spatial relationship between the mother cell and the 2 daughter cells? So if we have, say, an 8-cell embryo, we have 8 centroids in this volume. And then in a 16-cell embryo, those mother cells give rise to 2 daughter cells. Those daughter cells divide from the mother cell and migrate in different directions. And those different directions are characterized by an angle. So we can take the data, transform it, and get this secondary parameter. And then we have other properties which we can add to this model. So this is, an n-dimensional model. It's, as many dimensions as you want to put in here, as many things as you want to characterize in the point cloud. But basically, it's, it all kind of you can put this together, or take what you want and use it to model different aspects of the embryo. So in 2016, we talked a little bit about the OpenWorm Open House. This is okay. So that's a little bit about the data that we're using and some of the things that we did in our first 5 years. One thing I'd like to highlight from that first 5 years is the OpenWorm Open House. And this happened on October 25th, 2016. It's, the archive of this is still available. I was on the community committee at the time in OpenWorm, so we worked on putting this session together. What we had was, we basically had an introduction to OpenWorm, And then we had 15-minute presentations on major projects with an open worm. This was circa 2016. We also had some art for the project. So we had Matteo Farinella produce some artwork for open worm. And so that was featured in the meeting as well. So these 15-minute presentations kind of recap some of these projects, and some of them are very relevant still today, and some are less relevant. But those videos exist, and I'm glad we got them together because it was I think it's a very good collection or sampling of what's going on and what's required to build this virtual, nematode or virtual organism. So we have a number of projects represented here. C302, which is modeling the nervous system, which has 302 neurons. DevoWorm, of course. The educational initiatives with an open worm, which were, of course, distinct from what we do in DevoWorm, but we had kind of a talk on how to do education in this type of open source environment. We had SciUnit, which is a scientific replicability tool, a code replicability tool. Movement validation, which is measuring movement with a microscope of the worm and then validating it with machine learning. Cybernetic, which is, of course, the biophysics-based model I talked about earlier. WormSim, and Morphozoic, which, of course, is the cellular automata model. In 2018, then, we also had this Royal Society event. So this was an event that was held in London in person. The last event was held on the internet. We streamed it on YouTube. We had participants, ask questions and follow along. This was an event that was more sort of focused, not on the open source community, but on the scientific community, and people who could make it to London specifically. So there are a lot of British universities interested in C. elegans who attended, and I think it built some goodwill in that area. So DevoWorm, there's a piece on DevoWorm circa 2018 featured in this paper, OpenWorm, Overview and Recent Advances in Integrative Biological Simulation of Canarhabditis elegans. And so this is the poster here, Connectome to Behavior, Modeling C. elegans at Cellular Resolution. So this was, looking at sort of the connectome, going to behavior. So there wasn't a huge developmental component, but I think this meeting was kind of, brought together people. We could ask basic scientific questions in the context of modeling instead of thinking about this as a sort of exercise in emulation or high fidelity simulation. And so one of the driving questions that Steve Larson introduced here was, by simulating a small nervous system, which is C. elegans, it has 302 neurons, most in the head, but some in the other parts of the body, like in the tail or in the lateral regions. By simulating a small nervous system, can we learn information processing principles about how it works that we can then apply to the human nervous system? And so this is, something that people use C. elegans for as a model organism. But it's also something that if we simulate a very small connectome, we can learn about how connectomes work. We can learn about how motivated behavior works. And then we might be able to build up to what humans are doing in terms of neuroscience. We have sort of a neuroinformatics of the connectome at this scale, which is, of course, informative to what people are doing in human neuroscience. And so, it's useful for a number of reasons. And of course, simulating this adds another layer to it, because then you can say, what if you knock out certain neurons? What's the structure of the neuronal network? How does it work dynamically? So you can, if you have 302 neurons, you can simulate in a computer how a connectome behaves over time with respect to naturalistic behavior. And that's important. We can't really do that in humans. And so this is this is one of the driving questions that maybe one of the reasons why we want to simulate C. elegans in the first place. So this is very interesting. Other things we can do with this, though, is that we can take that connectome or a model of that connectome, a software model, and put it into a robot. So this is an example of some of the robotics work that was done in OpenWorm around this time, maybe up to about 2020. people were building these robots, which are wildly out of scale with the actual organism, but booting it up with some software, with the software connectome and driving the behavior of the connectome and the behavior of the robot. So this is, of course, a robot that can replicate some of the movements of C. elegans, some of the turns it can make, going back and forth, oscillating. And, they're very well-defined neural circuits in C. elegans, especially those that produce forward movements and backward movements. So you can actually plug in those small modules from the connectome, but you can also plug in the entire 302 cell connectome, which actually works sometimes to modulate these circuits. You can have a simple forward command, a simple reverse command, but you have inputs from the body, you have inputs from the environment that modulate that command and give you the type of movements you observe if you look at C. elegans under a microscope. So there's been some work done in robotics, putting the software into a robot's control system and seeing what happens. Now, there have been a lot of wildly speculative articles about this in the media, and I want you to put that aside when you think about this, and just think about maybe some of the more practical aspects of this kind of robotics research, which is to say that we can learn a lot about movement by scaling it up to a robot that we can observe, say, on a tabletop. Or we can even build a micro robot that would do this sort of thing. Just knowing, being able to interpret the inputs and outputs would allow us to learn a lot about movement. So, there are a lot of practical applications using robots, not using robots, but using computers, and so forth. We also have, the OpenWorm Movement Database, which is a database of microscopy data where movement is tracked, where a worm is moving across some agar, and you can see like the shape of the movements toward different points in space and things like that. And so this is something that you can look at both in the wild type and in defined mutants where they have movement deficits, where some neurons are knocked out, where they have metabolic deficits and so forth. C. elegans movement is not uniform. It's highly modulated, and the worm goes through phases of quiescence and activity, which are, important to characterize. If you're going to model the worm, you can't just model the ideals of that. This is an example of cybernetic, where a model of the worm is crawling through a simulated medium. It could be agar, it could be something else. And in reality, worms move through soil in 3 dimensions. They don't move, across the 2-dimensional surface of agar, which is a gel-like substance that's highly amenable to observing the worm's movement, but is not their native habitat. Their native habitat is soil with things like rotten fruit or other types of waste, and they go through the soil in 3 dimensions. So they go down, up and down, side to side, and diagonally. So there's a lot of complexity to their movements that we don't observe in some of these examples, and robots could actually replicate some of that as well. There's DevoWorm in the middle, and of course we had our role in this 2016 to 2018 period, and of course we've moved on from some of those things, but some of those other things persist. WormSim, of course, and then Geppetto, which is, of course, modeling the body and the neurons in the body, and doing a lot of this sort of neuroinformatics type style approach to modeling the worm. Now, in 2020, during the pandemic, I gave a talk at Artificial Life on sort of debunking a lot of the things that people have heard about OpenWorm, or some of the things you might think about simulating sort of a nematode. You might think certain things about how it's done, or you might have read about, booting up the brain of a worm into a robot, and, if that's a sign of the singularity, things like that. So I tried to sort of, set the sort of a realistic tone with this. It was interesting because I think it addressed a lot of problems that you might see in, any sort of artificial life endeavor, in simulating small organisms, small connectomes, etc. So OpenWorm is building the world's first virtual C. elegans, but how? Secrets will not be revealed, as it is open source, and of course we have no secrets in open source, at least not big secrets. And so this was an interesting talk. And then, this theme of giving talks in different places, this is very important, I think, to get the word out about things, about OpenWorm, about DevoWorm. And DevoWorm, of course, has been in a number of virtual conferences. We specialize in virtual conferences because we're virtual. We're a distributed collaborative group. And so in 2022, there were a number of talks given at venues like NetSci and Network Neuroscience, at the INCF Assembly, and the SciEM Workshop on Network Science. So we focused on network science a lot since about 2018. We've really tried to, think about development and the developing connectome as a network, and thinking about this very broadly, like what kinds of network science tools can we use to characterize the embryo? Well, it turns out we can use regular standard networks, complex networks. We can use neural networks. We can use hypergraphs, which are nodes within nodes. So we have hypernodes, which have a bunch of nodes in them, so we can nest our models. We can also embody our hypergraphs and embody our graphs into the body of like some biological system. So instead of just having like a hairball, which is something standard in network science, we can have something that resembles sort of a phenotype where there's polarity, where there's asymmetry, where there are connections that are functional, not necessarily just structural. And so in 2022, this is an example of the sort of virtual conference circuit that we participate in. So it fluctuates, over time, but we try to hit some of the main conferences and, in smaller events in complexity theory and network science and biophysics and so forth. This is a good map, though, of specific to DevoWorm, what our projects have been, like over from 2014 to 2024. So over that 10-year period, what kinds of projects have been going on at what time? And I only show this because we've had different foci over the years. And in summarizing this, I've noticed that some things have kind of tailed off. And I'm not really sure why they did, because they're good things, good projects, but they just kind of tailed off. We didn't talk about them much anymore. Sometimes that's because of the structure and nature of our group. We get a lot of people who come in and they do some work and then they have to do other things, they have other commitments. Sometimes there's students who have to drop what they're doing and go back to work or school, and sometimes people are doing them and they just kind of have other things they want to do. So, but since it's open source, so we can pick up a lot of the stuff and carry it on. So, from 2014 on, we've been working on this problem of differentiation waves in trees. So this is a problem of differentiation of cells into tissues, into different types of systems in a developing organism, finally leading to the kind of organism you might see as a nematode, where they have organs, they have sort of a symmetry, a bilateral symmetry, but it's asymmetric in terms of how the tissues are distributed. You have structure, but you don't have, it's not a totally symmetrical thing. There's differentiation involved. And we can characterize lineage trees as differentiation trees. We can characterize the collective activity of cells during differentiation as these differentiation waves. And so we've been working a number of papers over the 10 years of the group on that. Around about 2016 on, we've been working on developmental data science. And if you go back to that slide on the n-dimensional model of the point cloud, that's kind of the basis for a lot of our work in developmental data science. So this is where we take secondary data sets and we analyze them and, have publish insights from them. So there's a lot you can learn from existing data. There's a lot of data out there, not a lot of theoretical insight or, good models. And so that's been doing a lot of developmental data science for about 8 years of the 10. Of course, we did the morphozoic work that was early on, and it kind of tailed off in about 2017, 2018. But basically, those tools still exist. It's a cellular automata model where you have these nested neighborhoods. So it looks at the interactions of cells around a focal cell, and then that neighborhood has surrounding neighborhoods that it interacts with, and those interactions determine the states of the focal cells. So you end up with this system of sort of collective behavior where you can get waves, you can get fronts, you can get independent patterns that emerge. So you get a lot of pattern formation and a lot of larger scale structures that emerge. So it's interesting stuff, and there's a neural network component alongside of it that's really interesting. Um, I've been doing some work more recently on cellular automata. So at some point, we'd like to kind of pull this together. We've also have had and I haven't put it on there here, but we've had a longer standing interest in neurocellular automata. So that's something else that may fit into this area. And I haven't put it on here as cellular automata, but this has been kind of an off and on sort of thing in our group. So it's definitely, if you're interested in cellular automata, then please get in touch. We've also worked on 360-degree imaging and embryo viewers. So there were a couple of Google Summer of Code projects on this. This goes back to Richard Gordon's work on something he calls the Google embryo from like 1999 or something, where he envisions something like a Google Map system for embryos. So you can look at the surface of an embryo and you can get a sense of what, given your spatial location, what does it look like from different angles, looking at it in an immersive way. And so we've had some Google Summer Code projects that have addressed some of this using some data that we've been developing within the group. And so that's something of interest. We've also done diatom modeling. So instead of just focusing on C. elegans, we've expanded our work to single cell organisms such as diatoms. And there's a whole class of problems in something called single cell morphogenesis that's really interesting here. It's looking at single cells and colonies of single cells that, are very different from the kind of embryogenesis you see in C. elegans or in mammals or in other model organisms like Drosophila. And so looking at that, we can, again, it's a system that's tractable. We can get really good data. We can get really good models that are minimal and so forth. And of course, since 2018, we've been working on this developmental connectomes and embryo networks theme. And this is, again, applying network science in different ways to connectomes, to embodied networked systems in development. Then we have this machine learning theme, which is, of course, the theme that a lot of people are interested. They come to the lab and they're really interested in machine learning, but they're also interested in biology. And so, there's this stepping stone where you say, well, I'm interested in machine learning and I have the skills, but how do I apply that to biology? And it turns out you need to learn a lot of biology to really make that work. So to that end, we sponsored a course at the end of in the fall of 2019 called DevoWorm ML, which is the machine learning component of DevoWorm, where we focused on different machine learning techniques, deep learning techniques, and how it fits into developmental biology. Those materials are open source and they're available in our GitHub repository. But then that led us to something called DevoLearn, which is basically a pre-trained deep learning model for analyzing microscopy data of embryogenesis. And so we have really good data that allows us to look at embryogenesis. Is this dynamic process, segment cells, segment cell nuclei or cell bodies, and characterize each of these cells. And we have, in C. elegans at least, a way to annotate all of those data so we can build these highly annotated models. We can also train models in a way that allows us to take different data sets and easily identify those things. One of the things we've had a problem with in our machine learning work has been that it's hard to kind of characterize cells in microscopy images. Sometimes you have to do create manual masks to get some sort of training for the model. Sometimes you have to nudge the model along through in the training data set. And so these are things that maybe we can put aside if we can do like some sort of pre-training scheme. And so, we were working on this for a couple of years, and the outcome of this was this DevoWorm package that was published back in 2021. But we've expanded that to include various models, various neural network models, and more recently graph neural network models. So we're working on something called DevoGraph, which is kind of part of DevoLearn, and expanding our sort of approach. So we work with a lot of different types of model organisms. We work with nematodes, and this is an example of a nematode egg next to an adult nematode, or maybe a larval nematode, not sure. Then we work on diatoms, which are these single-cell organisms. They have silicate cell walls, and they have different shapes. They're very interesting organisms. They have a very interesting growth pattern. We've also worked with Drosophila data. There's all sorts of secondary data, not only on nematodes, but on Drosophila, on zebrafish, on the sea squirts or Siona intestinalis. These are all organisms that have good data for their development and their different developmental stages, but they all have different sort of modes of development. So the lineage tree in nematodes is different from the lineage tree in Siona intestinalis, and together they share this type of this mode of development called mosaic development. Drosophila has some mosaic development and some what they call regulative development, or where the lineage tree is less clear on what cells are going to give rise to other cells. And then zebrafish, which is even more regulative. So we have all these different types. We have cell tracking data for all four groups. We can build models from that. We can compare across those species. So we do a lot of comparative development. And then because we're interested in linking this to mammals, we do things with Mus musculus, where there is some good data for Mus musculus in terms of its early development in any case. But we have all this diversity of developmental systems because A, they serve as common model organisms, but B, they serve this comparative function. Susan Crawford Young has been developing some data in different really interesting ways on salamander embryos. So she's been doing a lot of work with building these microscopes, like what she calls a ball microscope, where you can take images from 9 different angles around the sphere of the embryo, and you can stitch those together into some sort of view that allows you to explore the whole surface, get different perspectives on salamander eggs. And so this fits into the model organism, and this fits into the comparative development theme, but it also allows us to just do some cool things with monitoring development and collecting data. And then finally, we have our in silico theory, which is where we can build artificial life models. We can do different types of simulations, exercises, and it tells us something about development that we wouldn't just know from doing a very standard plot of the data. So one of the things that we've explored is this theme of differentiation trees and mosaic development. We're looking at something called the differentiation tree, and we're looking at a mode of development called mosaic development. And so Richard Gordon worked out differentiation trees for organisms like salamanders, where you can look at the different tissues. You can look at different tissues as they split away from one another. You can collectives of cells that differentiate into different tissues, and it's largely based on signaling. So you get the signal that propagates from cell to cell, and you get this sort of, it leads to a certain lineage. In the case of a mosaic development, however, we already know what the cell is going to become, so it's no mystery. There doesn't need to be, collective behavior. It just needs to follow its program, or it needs to follow its sort of natural way of unfolding. So it's basically programmed into the genome. But what we can do is we can take a lineage tree, and we can analyze it. We can look at different properties of those mother cells and daughter cells and reorganize the lineage tree into something called the differentiation tree. And so what we can do is look at the relative size of each daughter. So we have a founder cell, which gives rise to two daughter cells, a larger daughter cell, and a smaller daughter cell. If we have a larger daughter cell, we move it to the left. If we have a smaller daughter cell, we move it to the right. Now, in a lineage tree, we usually have something, we organize the lineage tree along an anterior-posterior axis. That means that the mother cell gives rise to two daughter cells, and the daughter cell that's closer to the anterior end, or the head, is on the left, and the daughter that's closer to the tail or the posterior end is on the right. In this case, though, in the differentiation tree, we're sorting by size. So the larger cells to the left, the smaller cells to the right. And they aren't necessarily in that same in the same order as a lineage tree, but they can be, because it's basically just it's a 50-50 proposition, more or less. If the anterior daughter tends to be larger, then you don't have to reorganize it. And so we do this again and again over the entire tree. We end up with this tree that's reorganized locally. So we have these, we can summarize basically local division, expansion, contraction dynamics. So every time there's a cell division, a binary cell division, you look at the larger cell and the smaller cell and you sort them. And this describes an expansion versus a contraction event. So there's an expansion of cell size and a contraction of cell size, and these are all locally. And eventually, if you reorganize the tree enough, it might reveal that there are these larger scale expansion and contraction events as you flip those nodes, and that has consequences downstream. So we talk about this kind of thing in this paper, Quantifying Mosaic Development, that's in the journal Biology. So that's one thing we can do with lineage trees, is we can reorganize them, reinterpret them, find new information about development. We also did this for the Siona intestinalis embryo. What's interesting about the Siona intestinalis embryo is it's a bit more complex geometrically than the nematode. There's like this three-dimensional, it's a more complicated sort of set of divisions that occur. So it's not just, the lineage tree isn't oriented simply anterior or posteriorly. There's some other complexity to it. But anyways, you can create a differentiation tree like we have here. We have it sorted in the way that was described in the last slide. And for this early Siona intestinalis embryo, you get this type of differentiation tree. So stylized to show like the different, so there's this fourfold symmetry that emerges in this structure. You can see the four folds of symmetry, and then you can actually plot out what these cells are destined to be. So we know from the mode of mosaic development that these cells all lead to some fate, and it's deterministic. So we actually can look and we can classify them, and we can see that although there's fourfold symmetry, there's asymmetry in what these cells will become. So in some regions, you're going to have epidermal cells, and in the regions, a mix of different cell fates. And, some places they're just strictly mesodermal or endodermal. And so we have this sort of pattern, or even neural plate. So you can see that there are asymmetries that emerge very early on in what looks like a symmetrical embryo. We published this in Embodied Cognitive Morphogenesis, which is this Royal Society Interface Focus paper. We talk about a number of methods that we're using to look at development, ranging from differentiation trees to things more exotic models for looking at different aspects of embryogenesis. This is some of the newer work on cellular automata. So this is where we can work with these elementary morphogenetic structures, which is these one-dimensional cellular automata that are stacked in different ways to form patterns. Or we can use these nested morphozoic structures, which are two-dimensional. So you have this neighborhood here that connects to this larger set of neighborhoods here, and There are all sorts of nested effects we can examine, but we can also build very simple phenotypes. We can build these one-dimensional cellular automata and build sort of iterative structures and modular structures from those. So we have a open source community, and I want to focus on that because, of course, we're very much an open source organization. We're also a distributed collaborative organization. So we do manage a lot of our communications and a lot of our research agenda through different tools. And so it's important to recap where we are with that. So currently, and we didn't start with all these tools, they kind of emerged over time. We have a set of Slack channels. We have a Slack channel in OpenWorm, and we have a Slack channel in Orthogonal Lab, in those two different Slack teams. We have channels DevoWorm, DevoLearn, DevoGraph, and those are both in the OpenWorm Slack, and DevoWorm Orthogonal in the Orthogonal Lab Slack. And the reason for that is because they serve different communities. The DevoWorm Orthogonal is more geared towards sort of AI techniques and cognitive science and neuroscience techniques. DevoWorm is where we discuss sort of the main project, a lot of the developmental biology. And then DevoLearn, DevoGraph is specializing in this deep learning and graph neural network space. So we're able to draw and specialize in different areas. We also have a web presence. We have a Weebly site, which is the main site, and we have a GitHub IO site, which is a lot of the specialized information, things like our DevoZoo initiative, which features our data sets, our model organisms, and some other things as well. And of course, these resources always change. So the GitHub IO is since about 2020. The Weebly site has been up since about 2017. And before that, we had like a single Gitter channel, which was like a chat channel. And so we've evolved our community significantly over the years. We're also present on GitHub and Hugging Face, so we have open source data, we have open source software in those places. We have a DevoWorm GitHub alias, and we have a DevoLearn GitHub organization. So we focus on the sort of the legacy DevoWorm stuff, some of the biological modeling stuff, and then DevoLearn, where we have this stuff specific to deep learning and graph neural networks and other things like that. Then on Hugging Face, we have models specific to DevoLearn. And some of the models, we have different types of models there for simulating different aspects of embryogenesis. So it's not just a matter of simple segmentation of images. There are a lot of really sophisticated tools there. So Hugging Face is good because it's a community rather than GitHub, which is more of kind of a place to go to get things. And so, but everything's available in an open source way. Finally, our meetings are managed by Jitsi and YouTube. So we have a Jitsi space where we meet once a week. And then we have an asynchronous component on YouTube. So we record the meetings and we play them, we put them out, and people can watch them whenever. And that really does engage with two different communities of people. So, we're able to manage our community in multiple ways, multiple audiences, etc. We have a YouTube channel for DevoWorm, and that's a very successful endeavor. We have, this is the, our YouTube channel with different playlists. So we have a number of different playlists. We have a network science playlist. We have a lot of our presentations and posters. We've participated in a conference called Dynamics Days, which is a dynamical systems conference. We also have networks and DGNNs. Network science and networks are kind of separate, but this is a very big focus of the group. We also have morphogenesis and complex systems, so we have different lectures on that, different talks, different kind of ideas shaping up around that. We have our lab meetings, of course. We have our DevoWorm ML content, which is that course, which we'd like to return to because a lot of the content there is from 2019. We have some summer code-related stuff, and then we have some tutorials. So we have a lot of things up here that, kind of over the years that we've been around, And so it's very, sometimes it's very enlightening to go back in time and look at all the past content to make connections. So I encourage you to explore our YouTube channel. We've also participated in the International C. elegans Conference. We've done this 3 times. One was in 2015, which I went to in Los Angeles. In 2021 and 2023, Porig Gleason from OpenWorm attended in Edinburgh, and we've been a part of that conference, on a semi-regular basis. So we had in the 2021 and 2023 posters, we had a little square here. The 2015 meeting, we had our own poster. But anyways, it's just a summary of what we do and how we do things. And so, definitely we have those on the OpenWorm website. Check those out if you're interested. This is a community of C. elegans biologists mainly. So it's not computational people, it's C. elegans worm biologists. So it's quite a bit different from, what we're usually serving in our open source community. So it's just, another opportunity to get the word out and, of course, make connections with biological labs. We're also heavily invested in Google Summer of Code. So we've sponsored students ever since 2017. So this is all from our timeline. In 2017, we kind of, after that OpenWorm open house, we started to get interns from Google Summer of Code. And so we started to build this sort of research area in machine learning or developmental biology. We started with a project in 2017 on developmental machine learning and computer vision. We continued working on deep learning, pre-trained models, maintenance of said models, started to work on graph neural networks in 2022, and this year we're going to maintain and improve our graph neural networks models exclusively. So we've had a number of students. I'm just trying to count here. I think 15 or 16 students. We just got 2 new students recently for 2024. So we've had over 10 students. So they're interested in a lot of different things, and I think they've gained a lot. We have this very rich educational pipeline aside from like tutorials and courses. We also have mentorship, and so some of these contributors are in places today that maybe they wouldn't have been in if it weren't for DevoWorm. But definitely we have some impressive things going on here. So Mayok Deb, who was one of our DevoWorm students in 2020, he ended up moving on to industry and writing for The Gradient. He wrote a nice article on a brief comparison between human perception and multimodal LMs, or language models. Arnett Banerjee, who was our intern in 2017, he's now the GSOC coordinator for INCF, so he's one of the people in our sponsoring organization. So that's nice to have that connection there. And then Asmit Singh, who was a student, I think in 2020, he's now at Georgia Tech, and he's working in the computer science department there as a TA and as a master's student. So many of our interns from Google Summer of Code have gone on technical jobs and other endeavors to graduate school, to industry, wherever. And it's nice to have been a part of their formative years. And so, we have this rich tradition in education. This is highlighting the OpenWorm, DevoWorm education project. So this is something that sort of was pre-DevoWorm ML, and it focuses more on sort of developmental biology education. So it's the set of resources that we manage on GitHub, and we haven't really done a lot with it recently, but it's basically the outcome of this Open Leaders program. So I had to create a project and build some resources around that. So this is basically we're integrating all these tools, Markdown, GitHub, using HTML, MathJax, and then, kind of building a curriculum for people to learn, people to modify. It's sort of like if you think about Wikipedia meets maybe like interactive education, and that's how it works. So it's basically focusing on these topics but it's not as restrictive as sort of Wikipedia. It allows people to edit things or to collaboratively build a project. And so that's the idea here. And it's basically, something that is available on, you can go and look at the materials on GitHub. That also led us to this DevoWorm ML effort. So that was later in 2019. This is a 16-week course in how machine learning techniques can be used to analyze and model development. So again, this is hosted on our website. The archive of this is on our Weebly site. And we did was we had the 16-week course over the fall, and it kind of covered each week some topic in machine learning, but relevant to developmental biology. So in 2019, we covered a number of topics. Input data, which is very important. That would be things like microscopy data, but also things like numeric data from simulations, numeric data from cell tracking, and other things. We had a focus on this digital basilaria project. So this basilaria, or the single cell morphogenesis, we wanted to characterize basilaria from various public domain images, characterize the motion from movies that were in the public domain, and get a sense of how to characterize a movement using machine learning. And so we had some students present on that. OpenDevoCell, which was the precursor to DevoLearn. We talked about that, how to apply these sorts of techniques to a C. elegans embryo. We talked about pre-trained models in the abstract. This also led to DevoLearn, but this was something that we wanted to kind of cover as a concept. TensorFlow, tutorial on TensorFlow. So, we had a student go through and show how to configure a TensorFlow model. We had a focus on specific topics like computational pareidolia, which is where, we might be more familiar with the term hallucination, but pareidolia refers specifically to computer vision, where an algorithm will see a pattern that doesn't really exist. So it's like visually when you look at a wall and you see a pattern, maybe a face or a person's outline, and it's really not that. It's just maybe like a stain on the wall or something, but your perceptual system sees it as a pattern. So it's a false positive. And so this is something you see a lot in computer vision, and we talked about that with respect to some of the models that we use. We also talked about GANs, generative adversarial networks, and how you can apply that to development. We also talked about, the sort of the game theoretic roots of GANs, which is actually quite an interesting topic, looking at like the adversarial nature in particular as a game. We looked at reinforcement learning, Peter's rule, invariance, meaning like, what are our assumptions leading into our models? Is the world highly variable, or is there invariance that we can pick up on and build a model around? And then hierarchical temporal models, which is a certain type of modeling that's bio-inspired, but also, this is moving towards alternate types of models, not just machine learning, or not just neural network models, and regression models. And so then in 2020 and 2021, we covered some additional topics in our lab meetings. And so there's a lot in that archive on the Weebly site that kind of covers all these topics and where they are in video form. And, this is something, again, that I'd like to revisit. I'd like to maybe introduce some new topics. I'd like to talk about some other ways to package this content. So it's interesting to revisit. Then there's DevoLearn, which of course we've been doing since 2020. And, we've been building these different algorithms. We've also been building training data sets, and then building a pipeline to theoretical insights. So, we might take some microscopy images. As a picture of Susan's ball microscope that I talked about earlier. You put an embryo in the stage inside the ball, and then the cameras pick up images from different angles. And so you can get multiple perspectives on the same sample. This leads us to this type of an output where you have cells in an image, and you can identify, you might use stains like, fluorescent signals, you might just use the different features in the bright field image, and you sort of segment those things. You build a segmentation map from the input. Sometimes the input is very noisy, so it's hard to see the features, and so you have to build a segmentation map and use techniques to get that signal and separate it from the noise and the background. Once we have a segmentation map, then we can build a quantitative model like this model of cell tracking. And we can also build things like networks and topological data analysis models. And so this is sort of the goal of DevoWorm, is to go through this cycle from collecting data to extracting features to building quantitative models. This is an example from DevoWorm. Specifically, this is our pre-trained segmentation model. So you can see that you have this original, which is an image with we have markers of the edges of the cells, but it's very noisy. We can extract a segmented image, and this is just running through the focal planes. So this is why it's shrinking down. But you can get this segmented model from the original, and then a segmented cell with a centroid. So you can take a feature like segment the cells by their membrane, and then you can extract a centroid or a secondary feature just by predicting on the segmented part. So we can do this, and we can use this to pre-train the model. So in the top, we're pre-training with semantic segmentation. We're using this for C. elegans embryo. And we have semantic segmentation. We also have non-semantic segmentation. So, we can do this in different ways. We can use semantic information. We can also use something like segment anything, which is a model that Meta put out recently. And we've actually been working on a segment anything model called CellSam. And we've been just basically using like prompts to get the segmentation to work instead of semantic information. So in this case, you use a bounding box. You define the area, you find the feature within that, or you use a label prompt once you get, if you have the information there, and you can extract things out. So you say each pixel represents something, and then you can extract all the pixels that represent that thing and be able to do it much more efficiently. Now, these are just two different ways we can do this. There are other ways to do this, but this is where we are. We also have this embryo generator model. So this is a pre-trained GAN that can produce different types of embryos from cell tracking data. So this is where we can build a generative structure out of nucleus segmentation and feed it into a generative adversarial network so that we can generate different forms of embryos. This is, based on this idea of generative models of morphogenesis and developmental biology. So there's a lot of promise in using generative models to build these kind of quasi-developmental systems where you mimic development and you can answer questions in developmental biology much better than if you were to try to, do a standard analysis of the data. Then we have the lineage population model, so we can take images, segment cells, we can add semantic information about the cell's lineage, and then if we take movies and we add the data together, we can actually start to build models that predict the emergence of certain lineages and the number of cells in certain lineages. So we can actually predict this against a known lineage tree. So we can take our data from a noisy set of data with respect to the cells, segmenting them out into individual cells against the background, and we can take a lineage tree that's known for C. elegans, and we can predict how closely are we matching a known lineage tree. So this is, for example, this top one is A, which A, B, and the AB sublineage has a lot of cells in it. So this is going to be the most prevalent lineage. And we can see this is the example where this blue line is the lineage tree as it's kind of moving faster. But this is the lineage tree as known, idealized lineage tree. And then this orange dotted line is from the segmented data. So we're actually able to track this known data. And so you have these plateaus, which are where there's no cell division in that lineage. Now, in the generative model, or this lineage population model, we can extract data more continuously, but we still kind of match that performance. So we're able to really accurately predict a lot of these lineages, maybe not, in the way that it's configured in the lineage tree, but we can basically match it, which is what we want. So we have a pretty good handle on some of the lineage population dynamics. So, this is our DevoWorm Group GitHub page. We have a lot of different repositories pinned here. Please visit that if you're interested in some of the stuff that we're doing with DevoWorm. We have the DevoWorm organization, which is separate, and this is where we have our models, our CellSam, our DevoLearn, our DevoGraph, some of the models like Nucleus Segmenter and Lineage Population Model. They're also on Hugging Face, so you can look there and under our organization name for models. And so now we move on to sort of this elephant in the room, which is that if you're around for 10 years, you have a lot of technical debt in your organization. So there's always this changing pace of technology, both in biological experiments and computing. So you can see with our computing that we're always constantly improving our machine learning models. We're always exploring new avenues in our cellular automata models. We're always exploring new avenues in our network models and some of the ways we collect data. It's always changing. And so the knowledge that we built on, and even the communities that we build, can become outdated. And so, these are the twin pressures of scientific innovation and the changing pace of technology. So, if you visit DevoWorm, it's not all wonderfully organized and, perfectly understandable. There are a lot of gaps. And some of that is because we just can't keep up with the technical debt that we incur. And that's just a normal part of being a 10-year-old organization. So if you look at our docs and read the docs for the OpenWorm Read the Docs, the Read the Docs for DevoWorm kind of describe these different objectives. And some of these have, reflect reality in 2024. We do some of these things still. But some of these things, that's not what we're doing. And so this was done in 2018 and sort of the way that we envisioned things. Some of these things didn't happen the way we'd like. So we're not really focused on them. But nevertheless, this can be updated, of course. But this is just a good example of technical debt as it accumulates. We also have a lot of underutilized repos. And this happens when you start a project and then you don't really follow through, or you don't, you kind of put it as a marker and then don't revisit it because there are other things that intervene. We have a lot of underutilized repos on github.com/DevoWorm, so in the DevoWorm organization. And this is where we have various papers on lineage trees, differentiation trees, and spatiotemporal development, characterized in a couple of repos. So we have this repo reference space for embryos, which kind of is sort of wanting to go further in terms of the representations we use to represent spatiotemporal structures in which embryos are embedded in. Remember that n-dimensional model that we introduced early in the talk, but also the RDF model. Those are reference spaces, and those are used basically to organize the lineage tree, organize the differentiation tree into a spatiotemporal model. We also have the analysis of developmental trees, which is, of course, analyzing developmental trees of different types and multi-cell systems. Those are all things that we kind of built we put titles on, we put some things in there, but they're underutilized. And, the spirit of our organization, our group, is to sort of start off things and then have people pick up. And so sometimes, there are things that might be of interest to people. Sometimes we don't complete them to the state that we would like it. So that's a source of technical debt. There's this other paper that was written in 2020, so the pandemic year, at the height of the pandemic, called Developmental Incongruity as a Dynamic Representation of Heterochromy. And this is a paper on the bioRxiv. This features a number of tools that we haven't necessarily revisited as much as I would like. There are these triangular state machines, which are, these kind of We're sort of like a combination of a genetic regulatory network and sort of a motif in a lineage tree. There's also models of heterochrony, which is differential growth during differentiation and development. And so there are all sorts of different, in their developmental incongruities characterized by diapause phases in organisms like caterpillar to butterfly transitions and things like that. So there are all these different tools that are in kind of various degrees of kind of completion and things that we would like maybe to pick up later if people are interested in using them. And it's always a challenge in open source in terms of maintaining things and in terms of kind of driving things forward from a basic state. So this is something that you don't, it's not just our fault. It's something you see a lot of in academic labs and a lot of open source technical projects as well. Another thing we've done that, kind of drives a lot of this forward, but is necessary, I think, for really getting to the biology of it, is having these open questions that we pose. So these are open questions. Richard Gordon has submitted some open questions. He likes to do this from time to time. This is something that these were open questions that were submitted in December of 2023. So they're just things that maybe are pie in the sky. Maybe they're just kind of like, questions that you might ask if you're interested in a model organism that hasn't been explored very much, or maybe a prior piece of work that you published and you want to go farther. So, these are things that we do on a regular basis in our lab meetings. So an example of the open questions that Richard asked, the first one is, imagine the changing network of neighbors from the point of view of one cell in an embryo. So here on the right, I've built this model of a network where you have kind of the point of view of this end, and you're looking at your neighbors, and you have this perspective on your neighbors. And of course, your neighbors all have a perspective of all their other neighbors. So your neighbor to the top, they're going to have a different perspective on the network than you do. And the node in the middle, they're going to have a different perspective on the network. When I say a view, a point of view, I don't really mean like that cells in an embryo have eyes, but they do have sensory apparatus. So they can, do things like, make different taxis or directed movements towards resources, where they can get a read of their environment. And so, depending on where you're on the network, those kinds of conditions are going to change. And so this is something that, it's important when you think about networks to think about what the nodes are doing and their behaviors and how that translates into connectivity. So even from that kind of out there question, you can get a lot of interesting outcomes in emerging work. Another question he asked is, can we shift diatoms? And they could be more generic to mean cells, but can we shift cells, in this specific case diatoms, from one category to another with something he calls the compustat? And the compustat is a tool. It's kind of related to homeostasis and some of the cybernetics questions that he wrote a paper in the 1960s on this. And back then, of course, people were very interested in homeostasis, and they still are, but it's not something we typically think about in conventional biology. There are different ways that we can approach it as a computational group that might add some insight. So we can go from cells to these kind of systems of cells and how they're regulated. And, can we shift those cells from one category to another, or one fate to another, or something like that? And then, there are other things like pointing out that there are these areas of underutilized or underexplored biology. So there's this area which he terms the early development of Caecilians/Apoda, which is this worm here. And it's not C. elegans, it's just a worm. And basically, there's this literature on this organism from dating back to 1889. So this is something that, if you were a computer scientist, you would probably never encounter. But in our group, because we have this interest in developmental biology and sort of this theoretical biology, we would encounter this and be able to exploit that work and bring it forward. So, there's this Ichthyopsis glutinosus. This is the Ceylon blind burrower. So this is the worm over here, and sort of looking at that model system. And so this is another thing I think he wanted. He's kind of wanting someone to review it. But, this is something, again, it's a very obscure system. How do we bring that into the present and use it as a model organism? Then we kind of go over some of these open questions, and we kind of think about concepts. So back in July 2021, we had this set of open questions on what it means to be an environment for cells, for embryos. So, thinking about, like, if you're sitting there listening to someone talk about environment in a set of papers or in a discussion, you might ask a couple questions. You might say, first of all, what is it? What is its relevance? And how do you define it? So from that, we took apart this concept and kind of made 2 points here. The first is that environment is permissive. So it provides general information about changes, stresses, and intensities, and those are transduced or converted into the biological system. So it allows for change to occur. It gives information. The second is that environment is instructive. So this is where we have specific information, such as ratios, patterns, codes, which are transduced into the biological system. So we have this permissiveness where things cross the membrane and into the cell. It's general information gets transduced, but we also have specific information, which are things like ratios, patterns, and codes that also get transduced. So it goes from being permissive to being instructive. So when you break down a concept like that, it allows you to think about it in new ways, and it gives you an avenue to theory and maybe even to modeling. So I have to mention that we've lost at least one of our members. We know we get a lot of people coming in and out of the group, and Steve McGrew is a long-standing member, and he gave a really good talk on phytography back in 2017. And Steve did a lot of work on different things. He did a lot of work on computing. He ran a blacksmith shop in Spokane, Washington, and he was also an artist. And he died a couple years ago of cancer. So, in memoriam to Steve McGrew. And thank you for being part of our group for many years. So in the next 10 years, we'd like to tackle a number of themes. This is just kind of an incomplete list. So the first one is from that Royal Society interface paper. We talked about different models. We talked about differentiation trees. We also talked about something in terms of cellular and tissue tensegrity. And this is where cells will form tissues, but also form arrays, which are ultra stable. And so Susan Crawford Young is interested in this topic. Richard Gordon and myself are interested in this topic. And it's a really interesting topic because there hasn't been a lot of work done on sort of the computational modeling of tensegrity outside of like mechanical systems. So doing this in a biological system poses some interesting challenges. And it poses a new kind of approach to theoretical biology. We, of course, want to pursue more development of DevoWorm and developing the software there. We also want to model developmental dynamics and continue to do that and build better models. We also have a slight interest in early life, and especially in terms of ensembles and pattern formation. So this is where you have, macromolecules that emerge and maybe form single cells, and maybe even form populations of cells. But that self-assembly process, merging that with like sort of an ensemble approach from statistical mechanics and pattern formation, and finding the parallels between those is kind of an interesting thing for us, and we'd like to explore that more. We're also interested in differentiation waves, and this is a picture of a differentiation wave in the imaginal disc of Drosophila. So this is where we actually had this high density drawing, camera lucida drawing of a imaginal disc. And they gave us really good resolution as to the cells. We didn't have to segment them because they were already segmented by the artist. But we were able to take this image and then analyze it analyze sort of the frequency domain and the other aspects of this image to give us some information on how a differentiation wave propagates across this tissue. And so this is what they call the furrow with this black bar here. And that furrow kind of drives this differentiation wave across this structure, this imaginal disk. So you see behind the differentiation wave, you get a different sort of formation of cells than you do ahead of it. So looking at model systems like that, we can characterize these kind of theoretical systems. Morgan Huff and myself are interested in organoid modeling. So Morgan Huff is, he's had this long-standing interest in organoids as well as in neuroimaging and computational models. And so, this will be an interesting area where if we model not just the embryo, but these organoids that can be grown from stem cells or induced stem cells, induced pluripotent cells, to build structures that look like something you'd find in an embryo, but actually are not. They're organically grown, artificially grown. But you can actually model that growth process and differentiation process in different ways in those organoids, and also look at things like human disease and other types of things. But finally, we want to follow up on the network science and developmental graph neural network stuff that we've been doing all along. So we want to continue some themes. We want to engage in some newer themes and drive those forward. So our contributor over the years, we've had a lot of contributors. This is a list of contributors from the page on our Weebly site. I don't know how many are here, at least 35 or 40. So we have a lot of, the point is we have a lot of people who have contributed over the years. Some of those people have been Google Summer of Code students. Some have been core contributors that attend every week. Some people have come and gone. And so we want to thank all of them for participating in the organization, in the group. Thank you.